Nerve tissue. The nervous tissue is a main component of the nervous system. The brain and spinal cord and nerves, which regulates and controls body functions. It is composed of neurons, which transmit impulses, and neuroglial cells, which are sepotent cells, which assist and help to neurons, provide nerve impulse, and provide nutrients to neurons. Function of the nervous system are sensory input, integration, controls of muscles and glands, homeostasis, and mental activity. Development of nerve tissue. Development of nerve tissue is named neurallation. The neurallation is the formation of the embryonic neural plate and its transformation into the neural tube. The stages of neurallation. First stage, formation of neural plate. As cells of the neural plate can be distinguished as elongated cells in the dorsal region of the ectoderm. The neural plate forms the neural folding. The second stage, formation of neural groove. The dorsal edges of plate thicken, forming the neural groove. Third stage, elevation and convergence of neural folds. And fourth one, formation of neural tube. The neural tube gives rise to the central nervous system. Cells lateral to neural groove from the neural crest. Also derived from ectoderm, the neural crest has sometimes been called the fourth derm layer because of its importance. Neural crest cells give rise to a variety of important structures in the adult body. As neurallation proceeds and the neural tube begins to form neural crest cells, and a group of cells positioned on the top edge of the formation neural folds. One of neural tube has formed and invaginates the neural crest cells formed a distinct population of cells resting on top of the neural tube. Cells of the neural crest undergo migration and give rise to skin pigment cells, which are melanocytes, neurons of the dorsal root ganglia of spinal nerves, and the autonomic nervous system, both sympathetic and parasympathetic ganglia. And Schwann cells responsible for myelination of peripheral nerves, adrenal medulla, some bones and cartilage in lower jaw. Let's go to the main cells of the nervous tissue, which are neurons or nerve cells. The neurons consist of cell body or perikaryon, containing the nucleus surrounded by cytoplasm and processes, which are dendrites and axons. Dendrites, which are multiply elongated processes specialized in receiving stimuli from the environment, sensory epithelial cells, or other neurons. And axons, which is a single process specialized in generation or conducting nerve impulse to other cells, nerve muscles, and gland cells. All axons originate from short pyramidal-shaped region, the axon hillock. So that dendrite there are processes of neurons which get information from anywhere and send this information to the body, to the pericardium. And axon, it's a process of the neuron, it's always single, which get information from the body, from the pericardium, and bring it outward of the body to the work organ. Cell bodies are situated in the gray matter of CNS in sensory and autonomic ganglia outside of PNS. CNS it means central nervous system. PNS it means peripheral nervous system. And neurons vary variable in size from 5 nanometers in cerebellum to 130 in the anterior half of the spinal cord. They have a rounded oval, star-shaped or pyramidal shape, and nuclei of neurons are large, spherical, pale staining, and centrical placed with a prominent nucleolus. Cytoplasm contains nasal bodies, neurofibrils, Golgi complex, mitochondria, inclusions. The highly developed rough endoplasmic reticulum organized into aggregates of parallel cistern and ribosomes. Between them appear under light microscope as a basophilic granules called nasal body. So nasal body is a greatly development rough endoplasmic reticulum. Nasal bodies are present in cell body and dendrites. They are absent in axon. Neurofibrils stained by silver salts are cytoplasmic fibrils in the cell body where they form branching network and extend into all processes. Each microfibril consists of collection of neurofilaments. They provide mechanical support and stability to the cell. Golgi complex is well development perinuclear in position, and mitochondria are numerous in the cytoplasm and extend into all processes. What about inclusions? Inclusions of pigments such as lipofuscin are residual of undigested material by lysosome. Classification of neurons. We have some type classification of neurons. First type of classification, it's a morphological classification, is based on the number of processes. Unipolar neurons are a neuroblast. Second type, pseudo-unipolar neurons have a single process which then forms a T-shape. It looks like the first pseudo-unipolar. It looks like a single process, but begins from the pericardium is a single process, and later will divide it to two processes, to dendrite and the axon. So really it's a bipolar, but looks like unipolar. So we call it pseudo-unipolar. Location of the pseudo-unipolar neurons in sensory ganglia, except vestibular and cochlear. Third type, bipolar neurons have one axon and one dendrite. Location: the cochlea and vestibular ganglia, and olfactory neural epithelium, and in the retina. Fourth type, multipolar neurons have more than two processes, one process being the axon and others dendrites. Location: they are most common neurons of the human nerve tissue and nerve system. So they take place in the central nervous system. Perikaryones of multipolar neurons compose the gray matter of the central nervous system. Next type classification is a functional classification. Here you can see multipolar neuron with a lot of dendrites and one axon. A bipolar neuron, a pseudo-unipolar neurons begins as a single but it's divided to two axon and dendrite. And a functional classification based on the functional relations. A first type is a sensory. Afferent neurons are involved in the reception of sensory stimuli from the environment and from within the body. They are mostly pseudo-unipolar neurons. They cells body leon outside of CNS in sensory ganglia. So sensory neurons get information from receptors and always connect with receptors, and they always pseudo-unipolar, mostly pseudo-unipolar, and bodies take place in sensory ganglia. Interneurons establish relationship among the neurons, forming complex functional networks. Interneurons take place between sensory and motor neurons, and they create reflex arc, and they create organized relation between neurons. They mostly depend on the structural classification or morphological. They are mostly multipolar neurons. Third type, motor or efferent neurons, control effector organs such as muscle fibers and exocrine and endocrine glands. Motor neurons bring information, feedback from the central nervous system to work organ. Always connect with effectors. And the motor neurons depend on the morphological classification, mostly the multipolar neurons. Glial cells. Nervous tissue has no intercellular matrix, and glial cells make microenvironment, and they fill spaces between neurons. Function of glial cells. The main function of glial cells, they are supporting trophic, secretory, protective functions. And let's go to classification of the glial cells. Glial cells at the first must be divided to macroglia and microglia. Macroglia, there are oligodendrocyte, astrocyte, ependymal cells. And microglial cells, they are not differentiated, they are not divided to any, doesn't have any subdivisions. Let's start from oligodendrocytes. oligodendrocytes produce the myelin sheath. They provide the electrical insulation of neurons in the central nervous system. These cells have a few small processes that are located around the axon, take place around the axons. The same function of Schwann cells in the peripheral nervous system. So we have like kind of oligodendrocyte, which are Schwann cells, which always situated in the peripheral nervous system, because oligodendrocyte is a myelinated cells which take place in the central nervous system. And the next kind of oligodendrocyte, there are satellite cells of ganglia. There are flattened cells. They form a capsule around the each ganglion cells. So we have, in general talking, we have subdivision of oligodendrocyte to Schwann cells, and the satellite cells. There are like two peripheral kind of oligodendrocyte. The next type of glial cells are astrocytes. Astrocytes are star-shaped cells which profuse branching processes. They contain large pale round nucleus. Astrocytes with few long processes are called fibrous astrocytes. They are located in the white matter. And processes are attached to the capillaries by perivascular feet. And protoplasmic astrocytes with many short branched processes are found in the gray matter. Many of them processes are attached to pia matter. Some extend to blood vessels and they attempt perivascular feet. They are connected with blood and they get nutrition straight from the blood. So function, they have a very important function, get nutrients and provide them to neurons, which never connect with blood vessels. And they take an active part in formation of the blood-brain barrier. Next type of macroglial cells, ependymal cells. Ependymal cells are low columnar ciliated epithelial cells that line the cavities of central nervous system. Cilia of the apical surface circulate cerebral fluid around the central nervous system. The apical surface are also covered with microvilli, which absorb cerebral spinal fluid. Ependymal cells are CSF production cells. Within the brain's ventricles, population of modified ependymal cells and capillaries together form a system called the choroid plexus, which produce the cerebral spinal fluid. But a layer of ependymal cells has a large difference from simple epithelial cells, because ependymal cells layer doesn't have basement membrane. And the microglia is a small elongated cell with short regular processes. Yes, microglia is phagocytic cells that represent mononuclear phagocytic system in nervous tissue. They are derived from the precursor cells in the bone marrow. They are involved with the inflammation and repair in the adult CNS. Microglia act as antigen presentation cells. They secrete a number of immunoregulatory cytokines. Let's go to nerve fibers. Nerve fibers consist of axons, axial cylinders, enveloped by the special sheaths derived from the glial cells. Classification of nerve fibers is based on the structure of their sheath. Distinguish two types of nerve fibers, unmyelinated and myelinated. Unmyelinated nerve fibers, location as the adult mainly in composition of autonomic nervous system. They are characterized by low speed of carrying out of the nervous impulse. At about 0.5 till 2 meter per second. Unmyelinated nerve fibers are formed by deepening the axial cylinder axon in cytoplasm of the Schwann cell. The plasmolemma Schwann cell invaginates surrounding the axon and forms duplication, which is a mesaxon. That fiber looks like electrical cable, and it can be invaginate more than one axon. So we call this such type fibers, myelinated and unmyelinated nerve fibers, like fibers of cable type. Myelinated nerve fibers, location at CNS and PNS and characterized by high speed of carrying out of nerve impulse. Since 5 till 120 meters per second. In myelinated nerve fibers, the axial cylinder is surrounded with a myelin sheath, around of which the thin layer, including cytoplasm and the nucleus of Schwann cells, is located. The structure is named Neurolemma. The process of formation of myelin begins with the invagination of a single axon into Schwann cell. Mesaxon is then formed. As the myelination proceeds, the mesaxon rotates around the axon, thereby enveloping the axon in concentric layers of Schwann cell cytoplasm and plasma membrane. This myelin sheath is concentric layers of Schwann cell membrane. So really this myelin sheath, it's a lot of layers of duplication of cell membrane of the Schwann cell. And cytoplasm and nucleus. Which are named the Neurolemma, have peripheral location in myelinated nerve fibers. Between the Schwann cells, there are short intervals at which the axon is not covered by myelin. These points are nodes of Ranvier. This distance between two nodes is called an internode and consists of Schwann cells. The action potential travels by jumping from node to node. This mode of conduction is called saltatory conduction. This type of carrying of the neural impulse by saltatory conduction increase speed of carrying of the neural impulse. So myelinated nerve fibers, there are quickly fibers.